I'd like to invite the founder of FUMI Ingredients, Edgar Suarez Garcia, who will introduce us to his company. So Edgar, could you please join us and tell us more about how FUMI Ingredients is working on producing functional ingredients from microbial biomass, and how are you going to eliminate eggs from our food system?
And thank you very much for the invitation. It's really nice to see the lineup, and also very nice presentation for the previous speaker. So thanks a lot for that. My name is Edgar Suarez. I am a co-founder of a startup in the Netherlands called FUMI Ingredients. I have a background in bioprocess engineering, and it was during my studies that the whole idea of FUMI became a reality. So today what I want to do is to give a very brief overview of, let's say, the ideas that led us to FUMI Ingredients and how we think we can use microorganisms in a different way. And in particular, we have been doing this to replace egg from food products. So this first slide shows a very simple description of what traditionally had been happening with microorganisms. So how microorganisms have been used in different areas in industrial applications. So for instance, if we speak about food, it's very easy to see industries like wine, beer, yogurt, which make use of fermentation processes to deliver a product. But also when we speak about biofuels, in the case of ethanol, production of industrial enzymes, of organic acids like lactic acids, or the pharmaceutical industry. So those are traditional industries in which we see microorganisms being used. Now recently, and specifically in the alternative protein segment, we see precision fermentation becoming more popular, and also at a larger scale, and as well cellular agriculture. Of course, there are more applications, but let's say that this summarizes the main fields in which microbes have been used. But when you first have a look at the type of processes involved in microbial biomass, of course, you first start with a fermentation process. Then you go to a downstream process in which you, let's say, extract the compound of interest. And then in the end, you have a final product. And the first thing we identified is that normally this full process is really complex. And then you have many unit operations that you need in order to fully purify your product of interest. And this happened traditionally in industry, but it's happening also nowadays with new processes. You see that it's really intensive, the processing, in order to get a product with high purity in high quality. This is a picture that shows a typical DSP process for a certain biotechnological process. And you don't really need to pay attention to the details, but the summary that I want to give here is that it's rather complex. So in total, 32 unit operations. And some of these unit operations are not very mild. They involve the usage of solvents, or chemicals, or high temperatures, or pH variations. And in the end, that leads, of course, to very high OPEX and CAPEX values, but also often they decrease the functional properties of your product of interest. So during my studies, the very first thing I was having in mind is, how can we reduce the complexity of this type of processes in order to preserve functionality, but also to make the process more economically feasible? The second thing that at the time which we were starting FUMI Ingredients was related to the fate of microbial biomass. And when you look at most of the processes, so fermentation processes, you either have a final product to be the biomass, so either single cell protein or fungal fermentation. You can have a very specific product that could be either intracellular or extracellular. And we are speaking about a particular protein, a pigment, an acid, and enzymes. And in here, it also precision fermentation is included. So a very specific target protein that you need for certain application. But in most cases, what you end up with is a byproduct that is the microbial biomass. after you have extracted this very specific protein of interest, or you have extracted this pigment, the end of the process, you end up with a byproduct, the biomass. And when we were investigating what has been done so far with this microbial biomass, what we identified that is mostly used for feed applications. So you basically collect them and then you feed them to pigs or to cattle. For energy, so it's directly burned to produce some energy, or is disposed in landfills or other areas. So in summary, a lot of these processes produce tons of this microbial biomass, and often they don't have high value application. Mostly they have low value applications. So we started to think, can we do something different there? Can we develop technologies perhaps that allows us to make use of this biomass to increase the value, to maybe make use of them in other applications like food, but high value applications. So that was the second thing that was part of our discovery trajectory before FUMI ingredients. And the third one was about identifying what the microbial biomass is. So looking into the structure of the cell. And these are two pictures that you can easily find online or you Google it. But when you look inside, you discover this incredible complexity and this amazing structure inside of the cells. And when you look into the composition, you see that the cell organizes itself in a very clever way. And then you have some areas where you have some proteins, you have some areas where you have some polysaccharides, you have some lipid bodies, you have some pigments here, you have some acids. So it's all very specific compounds, but they're playing a function. They're, let's say, stabilizing the structure of the cells. So our line of thinking is, why don't we make use of this complexity inside of the cell, of these structures that the cells are creating and are stabilizing? And can we use these compounds in this structure in our benefit? Can we take advantage of the complexity of the cell to create new food structures? So that was our third question that we were having at the beginning. And then at the same time, we were thinking, where can we apply this? Where can we make use of this biomass and these structures inside of the cell? And then we started to see a typical plant-based products, like a typical burger. And we would buy it at the supermarkets, and then we would look into the ingredient list, and what we will find is a very long list of ingredients. And then you can read a lot of different type of proteins, different type of oils, there are starches, there are oils, there are other extracts. And then we start wondering, why is this list so long? Why is it needed to be so complex? Can we make it simpler? Because if you look at the typical burger made of meat, it's just meat, a bit of salt and some herbs to give some flavor, but it's a rather simple recipe. On the contrary, meat analogs, they're very complex. They have a lot of ingredients. So we started also challenging ourselves to think whether we can replace a lot of these long list of ingredients with more simple formulations, perhaps formulations that are based on microbial structures. So can we perhaps use the same oils that are present in cells and same polysaccharides that are present in cells and proteins? And perhaps we can combine it in a certain way to create these food structures. Can we perhaps use microbial biomass in a way that we can immediately mimic food textures? So all of these questions we were asking at the beginning in order to start with FUMI ingredients. So what is it that we do? So as FUMI ingredients, we actually developed the biorefinery platform. So we actually developed the process that is needed to transform the microbial biomass into functional ingredients. And of course, we want to do this in a very efficient way. So the process that we develop needs to be, of course, very simple, compact, efficient, and scalable. It needs to be also very mild in such a way that the products that we generate are still very functional, that can can be used in food applications, or for instance, dairy or meat, or other applications in which high functionality is important. And again, we wanted to move a little bit farther away from the typical idea of just providing a mixture of amino acids with certain nutritional value. What we wanted to do is to provide functionality, so binding ability, gelation, foaming, emulsification ability, that we can stabilize the food structure, that we can make a good texture that is juicy, that has the proper amount of moisture. So indeed, at the very beginning, we wanted not to use the biomass in the typical way that it has been used. On the contrary, we wanted to move to a kind of a new generation or a new way of using the microbial biomass. Also, something very important to mention here is that we were paying more attention to circularity. So looking into those industries that generate a lot of microbial biomass at low cost. And of course, the typical candidates there are industrial biotechnology, companies that, for instance, produce a lot of organic acids or enzymes, and they are generating these huge amounts of biomass that basically they don't use, that they just burn or dispose. Or the typical wineries, in which for them, the yeast biomass is a side stream, it's not really high value stream, or the breweries. So indeed, we wanted to combine all these elements, a very efficient process, high functionality, and also circularity. And this was indeed the foundation of FUMI ingredients. So in a short description, so we can take any type of microbial biomass, you name it, algae or yeast, bacteria, fungi. We have developed series of processes that we use to transform this microbial biomass. And we are particularly targeting functional applications. So indeed, it's not simply to provide a source of amino acids or fatty acids. We are trying to go into the high range of applications, so functional applications. The process that we developed is, of course, very robust, but it's also very scalable, because we are not making use of weird equipment or weird technologies. It's all off-the-shelf equipment that is easy scalable and has a lower capex in comparison to others. And again, emphasizing, very important, the high functionality. And with this high functionality, we can, of course, give high value, but also go for clean label. So reducing this very complex and long list of ingredients in a formulation, but trying to make a short and very concise ingredient list to deliver the same textural experience in food products. At the very beginning, of course, we started developing the technology, validating the product. And what we have seen is that regardless of the type of microbial biomass that we use, we always can deliver some fraction with very specific functionality. So we can target foaming, we can target emulsification behavior, and perhaps the most important is the heat set gelation. So this is a property that is very important, fundamental for making meat analogs, but also to replace egg whites, and specifically this ability to form strong gels under temperature changes. So when you heat it up and you cool it down to make a heat set gel. In addition to that, the other two functionalities, foam and emulsification, allows you to also seek for other type of applications. For instance, in the bakery sector, or if you want to go into more savory sauces. So all the ability of this ingredient to play in different areas of functionality made it also possible for us to start applying it to a broader range of ingredients and applications. So for us, it was also a great discovery to see that having such a low value biomass, we can transform it into ingredients that are behaving in a similar way as egg white, but maybe also not even as egg white, but currently people are using methyl cellulose, for instance, or other type of hydrocolloids to get to this type of textures, and to see that an ingredient that is sourced renewably from microbial biomass can compete with ingredients like egg white or methyl cellulose. Another was for us very important, and the starting point, of course, of FUMI ingredients. So again, as a summary, what we want is to make use of microbial biomass to produce food structures. This is more or less FUMI's approach. We go for less refined ingredients. So ingredients in which energy consumption is much lower. To produce it, energy consumption is much lower, but also the costs involved in producing this ingredient are lower, but also the losses. So the amount of waste that we generate in our process, much lower in comparison to other traditional ingredient production processes. We are upcycling rest streams. So typical streams that have low value in fermentation industry. And again, we want to go functionality over purity. It is not really necessary to go for 99% purity in order for you to have a functional ingredient. And that for us has made a really significant change, because that allows you to design much compact and cheaper production process, but also to preserve more the functionality of your target ingredient. And also to tell you a little bit about our journey. So it all started from my PhD in bioprocess engineering. From there is where we have all these ideas that I mentioned. Then we went into doing something with microalgae. So we started a little project called Alfie, in which we wanted to start playing with algae-based food ingredients. Then we joined the Accelerate program in the Netherlands as part of Wageningen University. And there we got let's say, the learning into entrepreneurship and how to really structure a company. And after all these learnings, by the end of 2019, FUMI Ingredients was born. So we have been active already for over 2 years. From a team of one person, then I have my co-founder, who is now in charge of more the commercial aspect of the company, our CEO. And from that moment, we have grown our team. So we are combining expertise in the food, like with the food scientists, nutrition, biotechnology, and process engineering. It's a very dynamic, very young team, full of energy and ideas, which is precisely what FUMI needs. So all these creative developments for new technology development. And something also about our current situation. So back in 2019, we were producing only some R&D samples, so in the order of 10 grams. Then in 2020, we moved a little bit into doing bench scale trials, just to produce over half a kilo, 200 grams. And then there we started to do some product validation with several customers that were interested in our ingredients. Then in 2021, and currently, we are performing several pilot tests in which we want to produce over 100 kilos. And again, this is to continue this journey with our customers, to really provide sufficient quantities for them to be able to test those in different applications, but also at a large scale. And we are in this journey together with several customers, and we expect that along this journey, we can also start a commercial operation that we can officially start selling our ingredients. So currently, our lead ingredient has been produced from yeast at the scale of 100 kilos. We have also done a few trials with algae successfully, but for now, we are mostly working with yeast. But we also expect in this year, in the coming year, to be able to work also with bacteria and fungi. And that's why we are also actively looking for collaboration opportunities with many companies in order to start sourcing our ingredients from different microbial biomass. What is very interesting is that we see similarity when we take different microbial biomasses, but also we see sometimes that certain microbial biomass gives a plus in certain functionality. So for us also will be very interesting to start collaborating directly with certain companies in which, for instance, we can use only bacterial-based ingredients or algae-based ingredients. And that will be one of the challenges and goals for the next years. And regarding applications, we have been doing mostly in meat analogs, but also working a lot in bakery, savory, and in other applications, which is really exciting to see. Just to give you some take-home messages from this presentation about the FUMI ingredients. So we are using, or we believe that we are using microbial biomass in a different way. We are not using it as a platform for production, certain molecule. We are not using it directly as a single-cell protein or directly put in a food product. What we are doing is transforming this microbial biomass for high-end applications, for functional applications. For that, we have developed very compact and efficient processes that allows us to transform this microbial biomass, but to do it in a cheap way, so with low capex and opex. Because of this functionality and the way we are producing this ingredient, we believe that we can offer clean label, so we can minimize the list of ingredients needed in alternative food textures. And finally, there are many applications in which we are active. Meat analogs, of course, egg replacers, but we believe this technology and the usage of microbial biomass can be even broader. So thanks a lot for the attention, and of course, ready to answer your questions.
Thank you, Edgar. We're all actually looking forward to seeing this journey and seeing where FUMI Ingredients is going to, when you're going to launch commercially. I think it's going to be very interesting to see on the shelves one day. And thank you very much for pointing out how much functional ingredients are important to the food industry. There was a lot of questions. The audience is very curious. One of the most interesting one is, how would you label these ingredients on a final product ingredient deck? Or else, how would you describe them on the label?
Yeah, correct. So it really depends on the type of microbial biomass that you are using. Because in Europe, we have a legislation, and let's say there are some microbes that have been used for a long time compared to other ones. So when you speak about, for instance, yeast, it's very simple because yeast ingredients have been used for many decades. So for that, you can label it as a yeast extract, or as a yeast protein, or as a yeast ingredient. When we speak about other microbial biomass that have been less used, or let's say only used recently, according to European legislation, then you need to go through a different procedure that is called the novel food applications. And in this procedure, with the food authorities, it is decided what type of information you have to provide in your label. So how you should label it. Our opinion is that in all cases, it is possible. I mean, after, of course, you go through the food legislation, it's possible to label it as a, for instance, algae protein or algae extract or algae ingredient, just to give an example.
Thank you. Thank you. Next, what are the main issues working with algae as a large-scale biomass material to turn into functional ingredients?
So the most important and critical challenge at the moment is cost, because the cost of producing algae biomass is currently quite high. Just as an interesting point, if you go to a winery, or if basically the yeast that they produce is I don't think it's a waste for them, but almost. So they almost give it to you for free, because for them it's just a side stream. But in the algae case, what happens is that the biomass is actually their main product. So it's really expensive. So very challenging right now is to decrease the cost of algae production. And the second challenge is the color, because a lot of applications or a lot of algae biomass are still green. So when you want to apply this, for instance, in bakery applications, people don't really appreciate a lot having very strong green color. So the second challenge I would say is to decrease the color of algae, and in some cases there are companies already doing that, producing kind of a golden or even white version of algae, which is really interesting to see.
Yeah, that's actually very, very interesting. Thank you. I'm sorry. Okay, how does the downstream processing for biomass fermentation differ from precision fermentation?
Well, they're actually quite similar. For instance, if you, I'm not an expert, so I only provide, let's say, my opinion. If you look at the traditional industry of producing enzymes, so there are many companies that, let's say, their business is to produce enzymes. So they need to, have the particular gene incorporated. So they use the cell as a production platform. They sometimes excrete the enzyme or produce it intracellularly. And then after many purification steps, they get this enzyme with a certain purity. And in precision fermentation happens in a similar way. You introduce the gene that you want to express, either a dairy protein or even egg protein. Then you ferment it, you either excrete it or intracellular, you need to purify it, concentrate it. So it's very similar.
Okay. What would you say were the biggest challenges of scaling up a process like yours? You said you're currently in the process of doing that. And actually, I have an additional question to that. You were mentioning 100 kilos. That is annually, weekly, or 100 kilos in general.
So our main challenge is actually having the funds to, because it's a really expensive one. The equipment that is needed is rather expensive one, and as a startup, of course, we don't have this type of capital. So having the proper finances in place to run these processes is actually a difficult one. So what we are doing now is that we don't own this equipment. What we go to a co-manufacturer, and a co-manufacturer is an institution or a company that they already have the equipment in place. So we pay them to produce a production run. So all these 100 kilos that I mentioned is by production run, by batch. So, and if we have, let's say, money only to pay one production batch, then so be it. So we run it, we have the 100 kilos, and then we use it. And if we have more money, then we can pay for more production runs. So this 100 kilos is per batch. And let's say one of the main challenges has been to get to a proper co-manufacturer that has the proper equipment that we need at the right scale and under the right conditions to run the whole process, and also to be food grade, a co-manufacturer that understands food industry, food ingredients. So that has been one of the challenges. But we found it, and let's say we are happy also with them.
Yeah, I'm very glad you found it. Did you maybe find any incubator program to build and analyze your business idea? How did it develop and grow?
Yes, so I mentioned it briefly in one of my slides, but we went through Start Life. And Start Life is the incubator of Wageningen University. It's also the university where I did my PhD. So it was also, let's say, rather simple just to go across the street to meet them. And it was a very interesting experience for me because I came out of my PhD and I had no experience at all in entrepreneurship or having a company. And I went there with an idea or with several ideas, and they were very kind to start guiding me. And then I entered their Accelerate program. So it's kind of an incubation program. And basically you learn, say, the basics, and you start connecting with other entrepreneurs, with companies, with investors. You learn the finances. So all this knowledge that you really need. And that for us also was a bit of a game changer, because as a only thinking with the scientist hat is not sufficient. You also need the hat of entrepreneur. So that for me was a very good experience with them.
Yeah, you need to have a lot of roles in order for everything to work. Could I please ask you to answer one last question?
Okay.
Which microorganism do you use for creating the strong gel structures you mentioned before? If you can share that.
Yeah, so the specific strain I cannot give, but we have done it with yeast. We have done it with bacteria and with microalgae already. And we are also doing some tests, or let's say looking to do some tests also with fungi, also to prove it with it.
Okay, perfect. Thank you so much for your presentation. It's actually been very, very helpful to hear about how important the functionality of ingredients are and everything we can do with microbes.
Thank you everybody.